I would like to welcome you. I'm happy you are around to concentrate 25 minutes on collagen peptides and how specific collagen peptides can stimulate cell metabolism and connective tissue. Well, 3 very small slides to introduce you to our company. Gelita, headquartered in Eberbach, Germany, is a $600 million company with 8 production sites worldwide. We are doing collagen peptides and gelatin since more than 100 years, and we are market leader with about 2,600 employees and 26% market share globally. Our company is basically situated all over the world, close to our customers and the raw materials. And here you see a quite nice picture from our headquarters in Eberbach, Germany, close to the River Neckar. The company is separated in 3 different business units. One is concentrating on gelatin business, food, pharma, phototechnical. Another one is on the mineral proteins and fats as a kind of side business for our core business. And finally, the collagen peptides for health, nutrition, medical applications. And we will concentrate today only on the collagen peptides, which is, from a more chemical point of view, a hydrolyzation product of gelatin, where we, in the case of collagen and gelatin, talk pretty much about texturizing, functionality, food technology, foaming, emulsifying. So it's to stabilize water and air, basically in food ingredients or in food products. This is what collagen and gelatin is about. But when you hydrolyze, cut down your protein to smaller peptides, you will end up in an arena which is called collagen peptides, where the collagen is getting biofunctional, where certain health effects are stimulated in your body, and I will concentrate my speech on this aspect. If you look in what we are doing, I already said we start from collagen, do hot water extraction, and end up with gelatin in most cases. But then the next step really is a very specific enzymatic treatment under very specific and controlled conditions to end up with different collagen peptides. Because it's very important to understand that we are talking about a name. Collagen peptides is a name for a group of products. It's not a single ingredient. It's not a chemical component. It's a group of different ingredients acting different when it comes to stimulation of certain cells. So we talk basically about the fibroblasts in the skin. We talk about the chondrocytes in the joints and the osteoblasts in the bone, which are the areas where most of your collagen in your body is existing. So in each of your bodies, we talk about 35% of collagen protein, where your body consists of. For example, in the skin, 80% of your dry skin mass is pure collagen. In your joints, similar level, ligaments and tendons, even higher percentages of pure collagen. And these cells are producing the collagen. For example, in the skin, the fibroblasts are producing new collagen. And we just found very specific optimized products which really optimize the stimulation of these specific cells to maximize the effect for beauty, joint health, and bone health. So we gave it a brand name, of course. We call the optimized product for joint health Fortigel, and for beauty from inside to maximize stimulation of the fibroblasts Verisol. And then we have, of course, a broad range of other proteins which are used for protein enrichments, which we call the Pepti Plus range. Let's concentrate first of all on joint health, and here I would like to give you a little bit of an insight into the mechanism which is behind these stimulating effects. The daily dose required is 5 grams, and you already see here, if you're familiar with the scientific arena a little bit, this is only half the size which is recommended by most of the published studies so far. And this is basically because of this very specific effect of a product which is basically optimized in the way of the existence of specific peptides. So we call it a MALDI-MS fingerprint, what you see on the right side, which really is what Fortigel represents, the existence of very specific amino peptide compositions, which can also be expressed in a type of very typical weight fraction diagram, as well as end-standing amino acids. The story of using these collagen fragments to stimulate your natural body functions is very old. Indeed, it's about 1,000 years ago when St. Hildebert from Bingen gave a recommendation that basically if you have joint problems, just cook a broth of meat and bones So what you do here is extraction of collagen from bones, and this gives you already a health effect which was known 1,000 years ago. All we do today is to bring this to an industrial scale and to optimize this effect and to substantiate it with science that we are pretty sure we will fulfill the EFSA claims very soon. So if you look at what the healthy joints are normally, it's a constant catabolic, anabolic, so upbuilding of new joint cartilage and degradation of old cartilage balance, which, as long as you're in balance, means that you produce as much new collagen as you reduce the old one, so you are in good shape. Under certain conditions, this system can get out of balance, so you produce more degrading proteins, enzymes, resulting in reduction of joint cartilage, which then results, of course, in osteoarthritis. This is a picture of what cartilage really is. I said already, high concentration of collagen, 25% of proteoglycans, so carbohydrates, and these cells embedded in this matrix producing, secreting the cartilage mass, which is a normal function of these chondrocytes. It's a production of collagen and proteoglycans. All we do now is to bring a certain fragment of collagen very close to these cells so that they are stimulated just to increase the production of collagen and proteoglycans, to result in a regeneration effect by increased cartilage mass. This happens because the cells are, let's say, simulated with a situation where the degradation is higher than it really is. In other words, these fragments of collagen peptides are perceived by the cells of increased degradation of body or material, which is why this stimulation occurs. The cell just thinks degradation is higher than it really is and improves or increases its normal working mode, which is also very important to understand. What happens as a result is, let's say, normal original material. It's not an artificial product which is secreted by the cells. So it's just the support of a natural product, a natural mechanism. For about 5 years, in cooperation with the Collagen Research Institute in Kiel, we did a lot of preclinical research to prove with really tons of data that all this is true and to understand why this happens and what the mechanism is behind. So it's really a requirement, if this theory is true, that there is a rapid absorption of the collagen material and not completely hydrolyzed down to amino acids, but that there is an absorption of partly intact material. Of course, a distribution and accumulation in the target organ, in this case, in this example, beneath the cartilage, and a stimulation of the chondrocytes. And I could show you, as I said already, a lot of these data are published, many more materials available, much more data that this all is true. For example, rapid absorption can be shown by an increase and decrease of the serum level of the marker protein, in this case hydroxyproline, to show that really collagen peptides are absorbed, not only as hydrolyzed material, but partially in intact form. So we can really see the UV diagram of collagen fragments, in this case exactly the molecular weight distribution of Fortigel in blood serum. And that's an experiment showing that there is a distribution accumulation in target organs. So what you see is basically 14C labeled, hot, marked peptide in comparison to just a hot proline, so 14C labeled proline. And you can see the accumulation of these peptides, but only in intact form, not the single amino acids, in cartilage, in this case of a mouse model. Of course, we can't do this in humans. So it's really an accumulation of the active material in the cartilage. And much more data about the stimulation itself, cell tests showing that not only collagen is produced, so for example here you see a treatment with and without Fortigel. Without, you see these small black dots, which are the cells, and the brown material surround, which is the secreted collagen material, cartilage material. And on the right side you see the effect of stimulation by Fortigel, and the same is true also for the proteoglycans, here green, and unfortunately you can't see the cells that good in this resolution. All this at the end, however, doesn't count, because everybody today is crying for studies which are double-blind, placebo-controlled, randomized, so PCT trials, and we have already a lot of them. Unfortunately, so far, most of them done in ill people, as EFSA announced quite lately that they are looking only in studies with healthy people, but we can go a little bit deeper in that. So we have about 2,000 patients running through these different studies, starting already about 30 years ago. Most of them are double-blind, placebo-controlled. And I would just like to show you one result, which was done in cooperation with the Harvard University Tufts Medical Center, published in Osteoarthritis and Cartilage in 2011, showing the effect of Fortigel, in this case here, on two different subjects, one in the placebo group and one in the Fortigel group. And very easily to explain, these red dots is unhealthy status, reduced density of proteoglycans, where green is positive. And this is very easy to interpret. It's just an indication that the status of your cartilage is getting much more healthier, significantly, when you use Fortigel supplementation in comparison to placebo for 10 grams. Interestingly, this study was not initiated by us or financed by us. The idea of this study really was to come up with a new methodology of analyzing your knee status, your cartilage status in vivo, where normally X-ray detection is used. And they just use Fortigel because if you want to set up a new study method, you have to be sure you have an ingredient which really works to prevent a false negative result. And this is why these guys took Fortigel to see if there is an effect. It can be detected with this new method. We have a new study around, not published yet, brand new data from 2012, where we already did with 20 studies and people with slight osteoarthritis, a reduction of the daily dose down to 5 grams with very positive outcome, significant effect after 3 weeks only. But this is non-controlled. And the next step for us was to initiate 2 more studies, both with 5 grams on healthy people, which are both designed to hopefully fulfill the EFSA requirements. So we did a power calculation upfront. We did declare a primer outcome and secondary outcomes. We did all our homework, and these 2 studies are running already. And we're pretty sure that we can hand in our second health claim, 13.5, very soon, for substantiation of this product in Europe. So what it is basically is the next generation of joint health treatment, where so far only anti-inflammatory effects with glucosamine chondroitin were scientifically proven. And we do have now many studies in people with osteoarthritis and increasing number of studies also with healthy subjects to show that we are really acting against the root cause of joint pains. It's not only a reduction of the inflammation, it's really an increased density, if you compare here, before and after treatment, an increased density and status of your cartilage with Fortigel. So it's a root cause treatment of the real problem of osteoarthritis. And of course, there are many products already around. For example, well known here in Germany, the Doppelherz product. That's from Aqua Pharma in France, for example. The latest launch of Sanofi Aventis in Mexico and South America. And many other products on the market already. Just as an example. So the marketing message here really is joint health and increased mobility, not only for people in the age of plus 50, but also for younger people more directed to sport, as this product here from Squeezy in Germany. The next product to talk about is Verisol, where the effect is pretty much the same. The theory behind it is the same. We're just talking about a different cell, fibroblasts in the skin. And therefore, we need a different ingredient to maximize this effect. And we do need a different dose, 2.5 grams in this case only, to substantiate the effect. To talk about beauty from inside, basically what it means is you can see a lot of topical cosmetic advertisement telling you that there is an increased stimulation of an area which is called dermis or epidermis. To understand what really happens, it's important to understand that there is an outside and an inside layer, basically, which is the epidermis outside and the dermis inside. And the collagen concentration of your skin really is dominating, focused here on the dermis with about 80% of dry skin mass. Aging means skin dermal collagen loss, which starts in the age of 30 and increases drastically after menopause and can be as high as 2% per year. So a change in the dermis where the epidermis basically just follows. So in other words, the root cause for wrinkles is taking place in the lower areas of your skin. Why is this important? Because with topical cosmetic, you can fill up those wrinkles, of course, but as everybody knows, it's just a very short-term effect. When the aging just continues, and by feeding the skin from inside, you can show a lot of information and consistency that this really helps to increase the root cause again. It's acting at a place where the wrinkles, where elasticity really occurs. And to act something with the root cause principle means also if you then use topical cosmetic, it just increases your status of your skin where you are. With regard to topical cosmetic, you can use still, of course, it's not to say that beauty from inside is better than topical cosmetic, but you're just on a different level of skin status. So you add something in addition, but starting from a different point. So the result is just better. And this all is, of course, as well approved by a mode of action which is shown already, but here with different data. So that's, for example, the 14C labeling of Verisol, and you can see the increasement again in the target zone, in this case the skin, and of course here a stimulation effect with and without Verisol of your fibroblasts. So all this is shown, but also here at the end only clinical studies count. And I just have a very small part of our studies results shown here. So we did 2 studies, in total about 200 subjects. First study was a little bit like a dose-finding study with 2.5 and 5 grams daily dose versus a placebo with about 60 women aged 35 to 55. And just to give you a very brief outline, we saw a significant effect appearing after 4 weeks already, which shows an increased elasticity compared to placebo. For 5 grams, a little bit higher by tendency, but 2.5 already works. We found significantly increased hydration, reduced dehydration, and all this appearing after 4 weeks only. The second study was then done only with 2.5 grams, so we choose a lower dose with about 110 women, and we took a look into wrinkle reduction, wrinkle volume actually, by laser light scanning, as well as the concentration of procollagen in the synovial fluid of your skin. And we see here, for example, an increasement against the placebo of 60% collagen in the synovial fluid of your skin, 60, which means this effect is not only significant, it's also relevant. And just to show you that how this works and how the before and after photos would look like in such a case, that's an example of the placebo group against 2 examples of the Verum group, the Verisol group. And if you don't see this well because of resolution or light, we can get a little bit into this data later on, but there's a very clear effect of reduced wrinkles and much more shiny skin, normal skin, higher elasticity, which can be shown or proven by this photo documentation. So what we have shown is that the oral administration of only 2.5 grams Verisol per day improves skin elasticity, reduce wrinkles, and increase dermal collagen production and elastin production as well, significantly after 4 weeks, and leads to higher skin tightness and improved epidermal barrier functions. All this takes place in the dermis, which means basically we increase just the mass of the collagen surrounding the fibroblasts to increase and push back this body-own material to prevent wrinkles and reduce it. So it's positively influencing epidermal and dermal barrier skin structures and counteracts skin aging and improves skin appearance. We did also a lot of market research to understand how this product would be perceived by end producers, by end consumers. We did 1 omnibus study, so quantitative research with 1,500 women in Germany, France, and Spain, as well as 6 focus group studies in 3 different countries. And we learned a lot, as you can imagine, from these 10 women sitting there in a room, and they really talk and discuss 2 hours about their impression of beauty from inside and what collagen could mean for them. But the most important point for me really was to understand that the term collagen as such is already pretty much known by consumer. So in the target group of women, collagen already tells them something. So I could name many different ingredients. If you go out on the street, nobody knows them. Here, people know collagen already. What this means, very easy, is you can do a nutrition content claim. You can name your protein, and this is already understood by the consumer, Article 8 nutrition content claim, and understood as something correlated with beauty. Beauty from inside is considered to be more effective than topical measures, as it works in a systematic manner. And this is also because of, mother's talk. Everybody knows mother telling you, drink enough water because it's good for hydration. Or stop eating so much sweets because it's bad for your skin. Don't stop smoking because it's bad for your skin. So it's already known pretty well that what you drink, what you consume has an effect on your skin. And this is just another ingredient which really uses this effect and brings it to a beneficial endpoint. And the requirement, of course, for any new ingredient is natural product, no negative side effects, which is fulfilled by Verisol. It's really a product which is very close to unlimited applications, so it can be applied in all these different products. It's clear water soluble, the dose is quite low, therefore the price point is only 3 euro cents, around 3 euro cents per day. It's very excellent clear water, and of course also warm water soluble, pH temperature resistant, and it has a very superior and very constant profile based on renewable raw materials. With regard to safety, IP regulatory status, very, very, very close. It's a food product, so it's not a food additive. You can do clean labeling. There are some patents around showing the significant increased effect, the optimized effect in comparison to other collagen peptides. And also already a patent application which is directed to cellulitis because of this increased elasticity in the skin. We have filed an EFSA health claims just some weeks ago, and you can see it on the internet page of EFSA. And with a little bit of luck, we have a feedback in the next 3 months, so close after Christmas. And as I said already, you can do your Article 8 nutrition content claim, talk about the protein and the enriched protein product, by 30% enriched protein product, and you can name it like we did here in this case and just call your product collagen water. And give it a nice female appeal. And water is also linked to hydration, so this is a very appealing concept, I think, where you can do nutrition labeling already today. Other products from the market, all with Verisol. Supplements are around, as well as instant food products, instant drink products. That's the chocolate. I like very much the idea of a healthy dark chocolate. This is on the Brazilian market from Snella. Vina milk. Of course, milk and beauty is also quite linked in some areas. So these are examples from the market. Well, and the last point to talk about, I have just 2 more minutes, is to say what comes next. So the next thing really is bone health, because the third organ where collagen is really dominant is the bone. And if you go back on the streets again asking people about what a bone is, they will tell you it's calcium. It's all about calcium. We need more calcium, we need the vitamins. And all this is true, even though I personally think we should consider much more how to get these calcium bioavailable. But that's another discussion. But at the end, if you look at a demineralized bone, osteoporosis status, you can see that there's really a decreasement of the total bone mass, which is not only inorganic calcium hydroxyapatite, it's as well 20% organic material, which is 95% collagen. And what functionality you have in the bone here coming from collagen is really making use of the strength, of the tensible strength of collagen, which is higher than steel, to give this composite material, what a bone is basically, not only the mineral density and strength of a mineral material, but also the flexibility of a steel-like material, which is in this case the collagen. You have the cells inside here as well, and the idea simply is to say we optimize the ingredient to stimulate the biosynthesis of the osteoblasts, of bone extracellular matrix, to act together with the calcium source and maybe the right vitamins, K2 and D, to increase the production of collagen in your bone to build up the matrix for the calcium to mineralize it or to participate on. Clinical studies are here ongoing. It's nothing to market today because it's not yet safe for EFSA. That's something which will come up pretty soon, I'm sure about that. Thank you very much, and if you want to know more about it, you can visit us at booth number 4F32. And if you have some questions, it's a pleasure for me, of course, to answer. Thank you very much.